Okay, so I think we are online. I'd like to welcome everyone to this next symposium, which is on the genomics of severe depression and electroconvulsive therapy. I'd like to welcome everyone. We have a great lineup of speakers here on this emerging topic of the genomics of a subgroup of depression. And my co-chair and I, Peter Zandi, would like to go with you through this session. So we have, and we'll introduce the speakers in a moment. We have Mikael Landen from the Department of Psychiatry from Göteborg speaking on a topic of the first GWAS, the larger scale studies so far on severe depression in ECT patients. Alessandra Minelli will talk about important aspects of underpinning biology of severe depression, and Alessio Squassiana, also from Italy, will talk about some specific telomere and biomarker empirical studies. And we're very fortunate to have the discussant, Dr. Pat Sullivan, with us today, who will try to knit everything together at the very end. So this is a program for the next 1.5 hours. Questions can be asked orally after the talks or through the chat box, so the Q&A box you can see there. Please write in your questions anytime, and we will try to address them as we go through the symposium. So that's a brief introduction, and perhaps we get started now with the first part. So the first talk will be shared between Peter Zandi and I will make a brief introduction to the ECT from a clinical perspective, as well as from the genomics consortium perspective, which I will explain. So I will give the clinical and scientific rationale for why we started to build the consortium of ECT genomics, and Peter then will take on from there, and we'll talk about specific experiences in the United States where there is a large proportion of the study and the consortium underway. So these here are my faculty disclosures. So just for people who are maybe not psychiatrists or not familiar with the idea of ECT. So ECT is a treatment for very severe depression. It is electroconvulsive therapy, which is provided to patients in very severe states of the illness. So unipolar or bipolar depression is the most common indication. And these epileptic seizures are induced, these epileptic fits are induced by the electric stimulation and relate then to certain clinical outcomes. So the outcomes are usually in many cases an improvement within a very short period of time after 1, 2, 3, or 4, 5 sessions. A usual course takes around 10 to 12 sessions of ECT, which then describes the whole course. So the neurobiology of ECT is not really well understood. There's so much happening in the brain during those ECT sessions. By the way, the fits only last for about, let's say, 20 seconds or 25 seconds in duration for each of the fits on a single day. And they might be repeated 2 or 3 times per week over 5, 6 weeks in an ongoing way. And during this time, the neurobiological changes are quite diverse, as you can see here, ranging really from neurotransmitter changes, neuroplasticity, functional connectivity, brain-derived neurotraffic factors. So there's so much happening that the exact biological mechanisms really remain unclear. There are a number of different predictors of ECT effectiveness. So as you can see here, for example, the severity of depression is important for our talk today. So the higher severity is usually related to a better response to ECT, as well as also to remission, and some very severe features such as psychotic features, which are indicative of very severe forms of depression, are also clinical predictors of remission. There have been some genetic studies, as you can see here, going back a few years on individual, so candidate genes. suggesting I don't want to go into the details, but as you can read here from the list of genes indicating those genes of systems, biological systems I was referring to earlier, having some response or efficacy with very diverse results, as you can see here, with positive or even negative results. And that kind of continues. Common genes for depression and pharmacoresponse are also showing up here, like the COMT gene, for example, in various studies with positive effects and prediction effects on response and remission. Dopamine genes also have been replicated a few times, but it's a very difficult area to derive the mechanistic understanding from these single candidate genes. And also others, as you can see, are more consistently negative, like the serotonin and G protein related genes. A number of have been investigated as candidate genes, but again, although negative here, this is an area which kind of has died, obviously, in the age of GWAS studies and needs updating to better methodologies and approaches. And one of the more recent approaches to this question was whether then certain genes and genetic load for depression, for example, would vary according to state of ECT patients. So this is a study which looks at controls, at self-reported depression, which is usually a milder form of depression, and ECT patients and their genetic load. And as you can see, that the polygenic risk score for MDD, for major depressive disorder, is higher, indicating a higher load for those MDD genes in ECT patients compared to controls, and also compared to self-reported depression, indicative of a perhaps different genetic makeup in ECT or severely affected patients. This kind of idea is also supported by recent findings from analyzing different phenotyping methods. So recurrent MDD, which is indicative of a more severe form, here shown to have a higher heritability than, for example, self-reported forms of depression, which you can see here on the right-hand side, and recurrent MDD on the left-hand side. So there's obviously some difference in genetic makeup based on the different subtypes of depression. So the open questions coming from this here is that ECT is a very effective treatment in clinical practice, but only a proportion of patients does respond, and a proportion of around 1/3 does not respond adequately. So one would like to know what a better prediction of treatment is, but also, as I mentioned before, understanding the underpinnings of the severe forms of depression from a genetics perspective is not well understood so far. So therefore, the rationale for the study and the consortium of genomics of ECT and severe depression has been formed here to better understand the genomic underpinnings of severe depression and ECT response in severe depression in comparison to milder forms of depression. So in 2017, a consortium was formed, and these are some of the founding members and still active members. As you can see there, we have published the methodology of the consortium in two major papers, one a longer version, which is in the European Archives of Clinical Neuroscience, and the other one here in Lancet Psychiatry, where the methodology is described. So it means that the consortium is existing. I mentioned before, from the rationale, we have two aims of the study, which is to study the genomic underpinnings of severe depression and a response to ECT. We'll talk briefly about assessment and inclusion criteria, which are very broad, basically all cases of severe depression and who receive ECT from unipolar or bipolar depression spectrum. So the goal is to recruit perhaps 25 or 30,000 subjects all over the world. And we'll hear something more about figures later on, and to enhance bedside to bench, but also bench to bedside practice directions. As I mentioned, the inclusion criteria are quite broad: age of 18, primary diagnosis of unipolar bipolar depression, and there is a decision being made that this patient should receive ECT. And then they can have depression with various types of symptoms, indicative of severe depression, catatonic or psychotic features, or without those. And there are some exclusion criteria, which you can see on the right-hand side, which are basically non-primary diagnoses of unipolar or bipolar depression, or having significant neurological or neurodevelopmental disorders. The ways of data acquisition is, again, quite broadly. We want to be able to capture various treatment settings, routine treatment settings, where either retrospective data collection is possible or prospective data collection. Retrospective, I think we will hear an example from Mikael Landen in a moment, from big registries, where you can go back to registered patients and analyze the response to ECT and the genetic underpinnings, but also prospectively of newly collected patients, which are then undergoing a collection protocol. The protocol is quite straightforward. It has a basic version looking at some minimum criteria, and then criteria as a minimal set for response studies. And there's also an extended version of the data collection form, which also looks at additional diagnostic and family background and so forth criteria. So depending on settings, different extensions of this version of data collection can be used. The questionnaire is available as an online questionnaire in REDCap. Many people know how this works, and this is a great way of collecting standardized measures. Obviously, but blood collection is an important part of it in order to collect the blood for DNA and GWAS, obviously. And this is already my second last slide, which indicates the collection sites, which are mainly based in Northern America, Canada, and the United States, in Europe, Australia, and Asia. We have around currently 50 research sites and around 6,500 patients which have biosampling completed so far. So I'd like to encourage everyone who is interested to become a partner of this consortium, and it's quite straightforward of how you can do this. So everyone who has access to these patients, who has interest in contributing to this consortium, and who's obviously the ability to collect some patients, around 100 or more during for the study period of a few years, are most welcome to participate here. And at this stage, I'd like to hand over to Peter to continue to talk about the experience in the United States.
Thanks, Bernhard. Yeah, so we want to talk about real briefly how we've been trying to contribute to this effort from the United States, especially in collaboration with the National Network of Depression Centers. I'm going to describe. Actually, sorry, Bernhard. Yeah, thank you. You have control of the slides. So we've actually been funded to support this effort to recruit patients for this large-scale global effort here in the United States with grants to Johns Hopkins and University of North Carolina with my colleague Pat Sullivan. Here are the aims for that grant, and they're consistent with the aims that Bernhard just described for the global consortium. We want to ascertain consent, phenotype, and biosample 15,000 patients here in the U.S. Receiving ECT for extreme depression. And then consistent with the global consortium, we want to combine this data with the data that's being collected worldwide to conduct a genome-wide association study of this extreme major depression to investigate the genetic architecture of extreme depression. And then finally, to conduct a genome-wide association study of response to ECT to see if we can identify genetic predictors of patients who are going to respond well to the treatment and who also might experience cognitive impairments. So next slide, Bernhard. And here is a list of the ECT centers across the United States that we've enlisted to work together on this ambitious effort. And in bold, you can see a number of ECT centers from the National Network of Depression Centers. We've been working together for several years to try to harmonize how we're studying ECT, and we've enlisted them and joined together with them, and I'm a part of that group to contribute to this effort. Next slide, Bernhard. The NNDC, or National Network of Depression Centers, is a network of 26 academic centers around the country and growing that provide care for patients with mood disorders, and has this tripartite mission to advance clinical care, research, and patient education. A number of the centers in the NNDC have ECT clinics, and we've been working together to try to promote best practices with ECT and to engage in multi-site research across the centers. Next slide. As part of our effort in working together, we've been trying to create a learning health system for extreme depression treated by ECT. This is a buzzword that's gained a lot of popularity in the United States over the last couple of years. And this is a figure that depicts the idea behind a learning health system. The notion is that we want to try to integrate clinical care and research in our healthcare systems. And as our patients are coming in for care, we're learning what works and what doesn't work and what's causing illness. And we're quickly translating that back into improved care. This idea that practice informs research and research informs practice. And a driving force of this learning health system is this idea of measurement-based care. And that's what we've been trying to implement in these NNDC centers. As patients are coming into our clinics, we want to measure how they're doing with their illness so that we can track and monitor how they're doing and use that to improve clinical care and to inform research. Next slide, Bernhard. So to build this idea of creating a learning health system for extreme depression treated by ECT, we've been working on 2 stages. The first is to implement a measurement-based care program, which we refer to as the Mood Outcomes Program. This is a program that we're implementing across all centers where we're trying to capture standardized patient-rated outcomes as patients are coming into our clinics, and we want to implement this at our ECT centers across the network. The second stage is to harmonize how we're clinically documenting ECT across the different centers so we can obtain standardized structured information about the delivery of ECT. And the hope is that this will help us engage in multi-site initiatives. Instead of everybody doing different things and capturing different information, if we can capture standardized information, it would help us work together. So this slide depicts the schematic diagram of the Mood Outcomes Program, our measurement-based care program in the NNDC. The idea, again, is as patients are coming into our clinics, we're capturing at every clinic visit these patient-rated outcome measures, the PHQ-9, the GAD-7, and the Columbia Suicide Self-Rating Scale. And these are being captured electronically at local sites, either directly into their EMR or through a central data system that we built for the NNDC. And the notion is that in real time, the results of those measures are presented to the clinicians to promote measurement-based care and to engage the patients and improve the care that they're receiving. At the same time, we're capturing that information electronically and sharing it with a central repository that is allowing us to use that as a foundation for multi-site initiatives across the centers. Next slide, Bernhard. We started the program in 2016. Now, several years later, we've been rapidly disseminating it to centers across the consortium, and we've engaged over 15,000 patients across our different clinics, and we're growing. Next slide. This is just a figure of a paper that we recently published to report on our work in developing this measurement-based care program to support this learning health system. And the notion and the hope is that we can take this program and start implementing it across each of our ECT clinics to be able to get standardized outcome measures as our patients are receiving ECT across the different centers. Next slide. At the same time, we were working on this other initiative. We formed a work group to talk and to make some decisions about how we wanted to harmonize the clinical documentation as we are delivering ECT. We formed a work group and we came up with a recommended set of core data elements that we propose that ECT clinics should capture at each ECT administration for good clinical care, but also to have structured information that can be brought together again across centers for multi-site initiatives. We've actually generated a flow sheet template that can be shared with different clinics and implemented in their EMR, electronic medical record systems, so that they can use that as a basis for documenting the clinical encounters. We're getting ready to pilot this flow sheet at Johns Hopkins and our sister site at Sibley, and then we want to rapidly disseminate to our other ECT clinics across the NNDC network. Next slide, Bernhard. And so this really brings me to the last slide. The idea is that we're building this infrastructure, the so-called learning health system, and our hope is that we can leverage that infrastructure to recruit patients efficiently who have extreme depression and are receiving ECT and can be a part of this global genetics effort. We are rapidly disseminating the program and working on it. And the idea is that as patients are coming into our clinics, all we have to do is consent them, collect a biological sample, and the rest of the data is already being captured as part of the clinical encounter. So the idea to facilitate the research process. So the NNDC is hoping to build on this infrastructure and excited to be a part of this global consortium and contribute to the Untitled Chapter. And so with that, the last slide for me is just a couple of acknowledgements to both thank the NNDC, Dr. DePaolo, the leader, and the folks of the NNDC ECT task group, and Irving Reddy, especially my colleague, and also Patrick Sullivan, my co-PI on this grant that we've been awarded, and of course Bernhard and the other colleagues of the international. With that, Bernhard, I'll end.
Thank you very much, Peter. That was, I just stopped sharing the screen, so we should be back. Thanks very much. That was a great overview. So we have some time for questions. There's one question here coming in the Q&A box, which asks, what about MDD patients with TRD referred for ketamine, not just ECT? So would they be suitable, perhaps from your perspective in the United States?
So we've actually been talking about this, about whether we should expand recruitment into the ketamine clinics. It is something that we're actively thinking about. Right now, we're focused on ECT, but it is a step that we will take. I'll just make one other quick plug. The same idea that I talked about for creating a learning health system for ECT, we're also trying to develop harmonized data systems for capturing data as patients are receiving ketamine through IV and esketamine clinics, with the idea that we'd be able to broaden what we're doing. So it's a great suggestion.
Yeah, perhaps also from a global perspective of the consortium, since we are collecting cases with severe depression, even if there are perhaps ECT is indicated, but they've never received ECT or are lined up for ECT but haven't had it and have had ketamine, for example, then these would be suitable for the global initiative. But as you say, you need to kind of, in the US, you need to sort of harmonize then the systems with each other because they're different systems, the ECT system, the ketamine clinics, so they need to speak to each other. That's certainly a challenge there. But globally speaking, I think in answer to that question, yes, one can include those patients globally speaking as well. And I think it makes a lot of sense to do that. I have a question for you if we still have time. I think we have, Peter. So the learning healthcare system. So is that something which, as you said, has started in a focus, like in your own hospital or university, and then it kind of spread out, or is it like a national initiative which you take part in?
So the concept of a learning health system is kind of a buzzword now, gains increasing popularity in the early 2010s. In the United States, as you know, the healthcare system is fairly fragmented across different systems providing care. So there are different initiatives going on to develop this idea of a learning health system in different places. We are at Hopkins trying to build infrastructure and capability so that we can become a learning health system in psychiatry, and this idea of expanding it across the network with the National Network of Depression Centers. But there are various efforts like this going on around the country. There isn't one national effort that it can point to, but it is an idea that's increasingly gaining currency across the country.
Yeah, fantastic. Very good. I think we are kind of reaching the end of time. Are there any more questions? Thanks to Francis McMahon to ask this question via the Q&A box. Are there any other questions coming in? If that's not the case, we probably move on. Peter, is that okay? Yeah.
Sounds great. Thank you.
Okay. So maybe I briefly introduce then Mikael Landen, our next speaker. And it's great to have Mikael speaking here from Göteborg to us today, because he will present results of a first large-scale GWAS study in severe depression and ECT, I believe. So we are very much looking forward to your talk, Mikael.
Thank you, Bernhard. Yes, so I will present the results from a genome-wide association study of patients with major depression treated with ECT. And these are my disclosures. Yeah, so just for background, for many years, genome-wide association studies demonstrated no significant findings for major depressive disorder, which was perhaps not surprising given its relatively low heritability estimates around 30 to 40%. But after this massive increase in sample size, a meta-analysis of 130,000 cases and more than 300,000 controls could identify 44 significant loci, followed by more than 100 significant variants in a discovery cohort of 240,000 cases and a replication sample of more than 400,000 cases. These studies began to shed light on the underlying mechanisms, such as altered gene expression in the prefrontal anterior cingulate cortex, and also a connection to antidepressant mechanism of action. However, the SNP heritability for depression is still low. In the PGC MDD cohort, it was estimated at 9%. And for a clinician like me, this might not come as a surprise. MDD is a large umbrella under which several yet to be defined disorders are likely to be subsumed, everything from mild symptoms that remit with counseling or physical activity to severe illness that with psychotic features and that might require inpatient treatment. But this quest for ever larger sample sizes in genetic studies have not only impacted on the depth of phenotyping, it has also required inclusion of presumably less severe cases bordering on normality in some instances. For instance, the case definition of depression for the large cohort from 23andMe was merely based on self-reports of having been diagnosed with depression. So what we thought back in 2012 actually was that an alternative to ever larger GWAS studies would be to focus on cases at the extreme end of the spectrum, which might increase statistical power. For example, a GWAS of subjects with very high IQ identified 3 genome-wide significant findings in a sample size of only 1,200 cases, whereas population studies with more than 18,000 subjects failed to do so. And with respect to depression, we have the Converge study that we're able to identify 2 genome-wide significant findings in 5,000 women with clinically ascertained recurrent MDD. And it also stands to reason that the clinical applicability is higher when more severe cases are included rather than people with symptoms within the normal spectrum. And you went through the ECT, the electroconvulsive therapy that has been used in psychiatry since 1938. It was then the Italian professor Ugo Serraletti and his assistant Luigi Bini first tried the procedure on a man. He had been found severely confused at a railway station and had been hospitalized for weeks without improvement, and he recovered completely after 11 sessions. Bernhard, you described what we use ECT for today. The main indication is severe depression. And importantly, ECT is not prescribed for mild depression, and the treatment therefore identifies the most severe cases of MDD, sometimes also therapy refractory cases. our study was christened Prefect, which stands for predictors for ECT. The overarching aim was to provide biomarkers to predict the effect and side effect of ECT, but also to collect a cohort of cases with severe depression for genetic studies. In this study, using the Prefect material, we tested if the extreme phenotype approach would be useful in psychiatric genetic research. Specifically, we aim to estimate the SNP-based heritability, and compared to milder forms, we also conducted a GWAS. So we ended up with, well, yeah, I should say that we used the prospective collection where we enrolled patients scheduled for ECT, and we sampled blood before the first ECT and directly after, and then at the end of the treatment. But we also ascertained cases from the Swedish ECT register, which is set up to monitor the quality of ECT. So we defined a broad case definition, which, for example, included depression in the context of bipolar disorder, that is bipolar depression, and a narrow case definition with unibipolar depression only. 2,700 cases met the broad definition and 1,800 cases met the narrow definition. 2/3 were female, 1/3 had attempted suicide, and cases had a mean of 2 previous ECT and 4 hospitalizations. So it was a pretty severe group. Controls were collected for the Swedish branch of ANGI, which is a genetic study of eating disorders. And we selected 2 MDD cohorts for comparison. First, the ICBT cohort. These are patients with depression who were treated with internet-based cognitive behavior therapy. The ICBT study excluded individuals with severe depression and also excluded individuals with moderate to high risk of suicide. And they also included only individuals who sought psychotherapy. And the second comparison group was the PGC-MDD cohort with a range of severity. And the genotyping on the Illumina GSA chip. And now here are the results. See if I can have a pointer. So we first found that the SNP heritability was 0.35 for the broad case definition, and 0.31 for the narrow phenotype. That is assuming a lifetime prevalence of ECT depression of 1%. And this here you see by contrast the SNP heritability estimates in the PGC MDD cohort ranging between 6 to 8%. This means that across a range of estimated population prevalences, the SNP-based heritability was much higher in cases receiving ECT than in the PGC MDD cases. We then looked at the correlation structure and found that Prefect cases showed significantly smaller genetic correlation compared with the PGC with respect to autism, neuroticism, and starting smoking. Prefect cases showed no association with alcohol consumption, coronary heart disease, or type 2 diabetes, which all correlated positively with a less severe PGC cohort. Finally, Prefect cases showed a negative correlation with BMI, whereas the PGC MDD cohort showed a positive correlation. So these are comparisons of polygenic risk scores. The narrow group is here and the broad group is here, the broad including also bipolar depression. And the narrow group had a slightly higher mean of polygenic risk score for MDD, but the 2 case sets differed more strongly in genetic risk score for bipolar disorder and also for cognitive traits, which might be expected given how those were recruited. Then we also, despite the small sample size, we conducted a GWAS with the 2,700 cases meeting the broad definition. And 3 SNPs reached genome-wide significant. And this one here was located in an intron of HLA-B in the major histocompatibility region of chromosome 6. And it had some LD friends. This region has previously been associated with multiple psychiatric disorders, including schizophrenia, depression, and PTSD. 2 other SNPs had minor allele frequencies near 1% and no LD friends, suggesting false positive findings. And then for the narrow case definition, there was only 1 SNP that reached genome-wide significance, but it had a minor allele frequency close to 1% and no supporting markers in LD. So it's likely a false positive finding. So taken together, our findings show that the polygenic prediction of severe MDD is not only stronger as apparent from the high SNP-based heritability, but also different as revealed by the genetic correlation structure and also genetic risk score analysis compared with the less severe cases in the ICPT cohort and the PGC MDD cohort. This is encouraging because it suggests that larger studies focus on the severe end of the clinical spectrum have potential to have more power to uncover the genetic basis of MDD. And yeah, the sample size is small, so it's great to hear about this international consortium of genetics of electroconvulsive therapy. And finally, I just would like to acknowledge all the co-authors, both on this presentation and upcoming publication, hopefully, and also the data collectors that did a great job, and the ECT units at hospitals in Sweden, and also the Swedish National Quality Register for ECT. Thank you.
Thank you very much, Michael. It's a fantastic study and really, as you said, encouraging results in terms of following on that idea in larger sample sizes as we try to achieve this with the genetics consortium study in ECT and severe depression. Let's look at, I just want to encourage everyone to ask questions, and while maybe people think about it, I have 1 or 2 for you, if that's okay. So in the beginning, you said that you were having a mix of cases with prospective and retrospective assessment, if I understood correctly. What is the proportion between the two, roughly?
I think we had around 800 prospectively collected and 3,200 retrospectively. And why that number is higher is because for this study, we excluded patients who got ECT for a manic episode, for example, and some other indications.
Mm-hmm. Okay, good. So, I mean, the numbers are too small for or too unequal for comparisons, but I just wonder whether there's any difference in the case definition, because the case definitions seem to make a difference between narrow and broader in terms of the GWAS finding in this relatively small study, whether the prospective or retrospective assessment might vary across the case definition. Do you know that just by chance?
Well, I don't know because we haven't checked honestly, but we used the quality register for ECT to collect the phenotype data in both cases. So it's the same system is being used for the prospective and the retrospective collection. So it should be could be the same.
Yeah, great. So there's a question from the audience, and you might have said this, but I will pose the question to you: whether you're also collecting RNA for dynamic measures at multiple time points, because you were looking at different time points, weren't you?
Yeah, Yeah, should have done that. No, serum and plasma. We collected serum and plasma, but
okay.
not, or no.
Yeah, yeah. Can I ask you another question, if that's okay?
However, we
yes.
did collect. We did well, of course, we did collect blood. I mean, you can look at epigenetics effects
yes,
during the course of ECT, of course. Yeah.
Yes, that would be very interesting to do. Yeah. One of the key elements of the study design was to compare the severe cases to the milder cases, and you had the IBC cohort with the psychotherapy was provided to these patients. The PGC was the other comparison group. How did you ascertain mild or moderate state of depression in the PGC, or was it just given the entire PGC has a mix, so you assume there was a lower severity? How was that done?
Exactly.
The letter? Okay.
Yeah.
Okay. Yeah, I think it's very interesting to also see the HLA region coming up here again. I mean, you mentioned schizophrenia. It's a few years back now where the complement, the C4, was identified then. But also in other treatment response studies, the HLA region does come up, like in bipolar lithium treatment response genetics. That's a region where it also comes up. So it's an interesting area, but it's, as you say, it's coming up, but maybe not too strong so far. Yeah. How would you value? I mean, you mentioned they had some SNP friends there.
Yeah.
But did you follow that up in any way with some functional analyses also?
No, we didn't. Maybe Pat will comment on that, but we were really careful. That was almost an add-on analysis given the sample size here. So it's good to see that we found a significant findings, but we didn't do much more with that than that.
Okay. Very good. Excellent. So I'm looking at whether there are any more questions from the chat room. There are no more so far. Any other questions? People want to ask verbally, orally? If that's not the case, I thank you again, Michael. It was a fantastic presentation, very encouraging results. Really, really, really interesting.
Thanks.
Thank you. So we move on to our next speaker, who is Alessandra Minelli from Brescia in Italy. So it might have been in your country if Corona and COVID would have permitted. But it's great to have you here today. University of Brescia in Italy, Department of Molecular and Translational Medicine. Dr. Minelli will be talking about giving an overview, I believe, on molecular biomarkers for ECT, what's present and where it might go to in the future. Looking forward to your talk, Alessandra.
Thanks, Bernhard, and good evening to everybody, or good afternoon, depending on where you are. During my talk, I will try to give you an overview about this issue, about the literature of this issue. When we speak about molecular biomarkers and ECT, we speak about studies that try to find biomarkers for identifying biological mechanisms underlying ECT, and also studies concerning predictive biomarkers in order to identify patients who are more likely to benefit from ECT, or if not, to have the opportunity to plan in an early another kind of treatment. The current scenario of literature is only on studies with a candidate gene approach. Few are genetic studies and most are biochemical one. Since are all study with a candidate gene approach, all the studies start with the investigation of system concerning the hypothesis of vulnerability to depression. So serotonergic, dopaminergic, and glutamatergic system were investigated, but with few genetic studies, with a small sample sizes and methodological bias, as already seen in the previous talk. there are sparse results, there are negative association mainly with serotonin-erging system, positive association with some polymorphism of dopaminergic system, but in general, the results are a few, not conclusive, and so we have no strong evidence of association with these systems. Other studies investigate instead immune and inflammation system. Here I summarize the results. So studies showed an increase in protein levels like cortisol, interleukins 1 and 6 immediately following an ECT session. And instead seems that ECT, after all the treatment, there is a reduction of these kind of proteins, but like cortisol, interleukin 6, anti-NF alpha. So we can say that there is an acute stress reaction of our body after once at the beginning of ECT, and instead at the end there is a reduction, but the data are not sufficient for understanding if this reduction is just a reduction of the stress reaction, or instead there is a reduction also in correlation to decrease in symptomatology. So the problem of the studies on this kind of system are that all are biochemical studies and are few and quite relative old with small sample sizes and methodological bias. Most studies concerning molecular biomarker and ECT were carried out investigating neurotrophic factors. Few genetic studies were performed with some contrast, some interesting but not contrasting results, and to date are very few data. Instead, most are biochemical studies, more are on BDNF, but not only, also on other kind of neurotrophins. And on BDNF, there are a large number of studies that lead to some systematic review and meta-analysis. One was just published, and all this data lead to the conclusion that BDNF increases after ECT, similarly with pharmacological intervention. All this data coming from BDNF, but also from data on VEGF, on other protein like S100, lead to one conclusive finding that ECT induces a neurotrophic effect. This is one of the few conclusive findings that we have in this field, but I think that is extremely important because ECT was considered for a long time treatment dangerous that can provocate brain injury. And this data, in my experience, my clinical experience, was extremely important also for change the idea against ECT, because maybe as many of you know, Italy, in Italy was invented ECT, but in Italy there is a strong opposition against ECT, so it's very difficult to perform in hospital. So if we increase the data concerning the mechanism, we can change the idea in patient and physicians. All this data lead also to this question if neurotrophin may be predictive of ECT response. But the data are not conclusive because there are no clear conclusion about the correlation between baseline BDNF and clinical response to ECT, because also, if you check this, for example, meta-analysis, the conclusion are different because depend of the study included, because not all the results are congruent, and also depend when is considered the time point for the response evaluation. Instead, Concerning VEGF, our group some years ago demonstrated a correlation between baseline VEGF and response to ECT. The study was a pilot study on a very small sample size. So some year later, we repeated the experiment in another group of patients, in a larger group of patients, and also in comparison with patients treated only with pharmacological therapy. And we found a strong correlation between the VEGF at the baseline and the response to ECT. In particular, the patient with a very low level of VEGF has had a very high probability to not respond to ECT. Our explanation is linked to the mechanism of VEGF, because the VEGF increase the vasopermeability through the blood-brain barrier, so augment the cerebral concentration of antidepressant in the brain. So it's like say that ECT increase the VEGF and VEGF helps to work the drugs. Except this kind of results that are interesting, but obviously it's necessary to confirm in other groups and a larger sample. Other kind of experiment concerning other kind of molecular biomarkers like microRNA, transcriptional study, epigenetic studies are extremely at the beginning. There are only 1 or 2 studies for each of these molecular biomarkers with no conclusive result. Sometimes it's negative, and the sample size are extremely small. So my conclusion are that at the moment, the molecular biomarkers of ECT and literature about that is that most are biochemical studies. A large number of studies are relatively old, with small sample sizes and methodological bias. Genetic studies are few, with sparse results and no conclusive findings. Genomic of ECT is not known because to date there are no GWAS published data. And data concerning RNA, microRNA, epigenetic, and so on are extremely rare. So the scenario is at present concerning this issue, we can consider it not good and in a depressed situation. But as you can see, and as already said also in previous talks, concerning the genomic of ECT, and also from the talk of Professor Bauen concerning the International Consortium of ECT, I hope that in the future we can have more positive findings and more positive research on this issue. I thank my group of research of Professor Generelli at the hospital in Verona, where we can still do ECT in Italy. Thanks.
Thank you very much, Alessandra, for this great overview. Although, as you say, it's kind of a little bit depressing, you know, because of the contradictory and non-conclusive molecular findings. So, could you perhaps, you know, looking at these different studies, also suggest where the error is? Is it just, you know, for the negative findings? Is it just sample size, or design of the studies, or any particular things you would like to highlight here?
Mainly is the sample size, because the genetic studies carried out to date are with sample size with less than 100 of patients, so it's a very, very small for genetic studies. And also that in general, the polymorphism study are the most famous, but maybe are not so involved in ECT mechanism.
Okay. But perhaps also there's another question. Do you think that the increased serum BDNF has anything to do with the brain chemistry?
All the
serum BDNF, yes.
The literature has shown that any kind of neuro stimulation treatment, like also TMS, TDCS, and so on, increase BDNF, and also other kind of treatment like CBT. So it seems that I think that is a neurotrophic factor of the treatment, in particular for neurostimulation treatment.
Mm-hmm. Yeah, okay. Very good. Are there any other questions from the audience? I'm just looking at the chat. I think that question was just coming from the chat. So then I think that's good. Thanks very much again, Alessandra. And we will move on to our next speaker, and that is Dr. Alessio Squassiana from the University of Cagliari. And he will talk specifically about leukocyte telomere length and response to ECT treatment. So a zoom and focus on a particular potential mechanism. Alessio.
Yeah, thank you, Bernhard, and good afternoon and good evening to everybody. So as already said, my presentation will be about some findings from two studies that are related to each other on two different sample sets. One about telomere length and inflammation in severe mental disorders, where we also explore treatment-resistant depression, and another one specifically focused on response to ECT in treatment-resistant patients. So as we know, aging is physiological, of course, but it's also a primary risk factor for several human pathologies. A large body of data suggests that patients with psychiatric disorders have features that support accelerated aging, and especially increased frequency of age-related conditions, and in particular those that are characterized by an inflammatory component, and especially cardiovascular disorders, but also metabolic disorders, which also greatly account for the excess mortality and decreased life expectancy observed in severe mental disorders. Several studies also report alterations of molecular measures that are usually associated with aging, such as increased inflammation and increased brain age, cellular aging, and short telomeres. So telomeres are highly conserved complexes at the end of chromosomes, and they ensure chromosome stability. Telomeres physiologically shorten after each cell division until they reach a critical threshold, below which they undergo undergo growth arrest and enter a senescent status, or begin the apoptotic processes. Different stressors, including chronic disorders, but also oxidative stress and inflammatory processes, can significantly accelerate the physiological telomere shortening. So reduced leukocyte telomere length has been reported together with increased inflammatory load and levels of inflammatory markers in major depression, schizophrenia, and to some extent also to bipolar disorder, even though bipolar disorder findings are less congruent. The main reason being that most of the studies overlooked the effect of treatment with lithium. As I will show you in a while, lithium has been significantly correlated with increased telomere length. So why leukocyte telomere length? Because telomere length is easy to study in leukocytes, it's cost effective, and it has also been shown that telomere length in leukocyte well correlate with telomere length in all the other body tissues. There's also data suggesting a correlation between pharmacological treatments, and specifically telomere length are shorter in non-responders compared to responders to antidepressants, even though this is based only on few studies. And as I said, lithium treatment correlates with longer telomere length. The role of telomere length in treatment-resistant depression has not been explored yet, even though several studies reported for another potential marker of aging, inflammation, increased levels of inflammatory markers in treatment-resistant patients. And to date, only one study explored telomere length in TRD patients treated with ACT, but reported no significant difference in telomere length between responders and non-responders. So we performed two different studies. The first one, as I said, is the aim was to explore the role of telomere length and inflammation in psychiatric disorders by exploring three different groups of patients, mainly major depression, schizophrenia, and bipolar disorder, and a sample of non-psychiatric controls. The second one was specifically focused on treatment-resistant patients treated with ECT and divided in responders and non-responders at different time points, as I will show you in the next slides. So for the first study, this is the features of the sample that we included. So it was 40 bipolar patients, 41 schizophrenia, 37 major depression, and 36 healthy controls. These are all the features that we tested in terms of correlation with telomere length or inflammatory markers. And the 3 that are in the red box were those that were included in all the models. So the models were controlled for gender, age, and BMI. So we measure telomere length using quantitative fluorescence in situ hybridization, which is a very sensitive technique, but it's also very time consuming and expensive. And this is one of the 2 reasons why the sample size is not very large. The second reason is that we applied very stringent inclusion and exclusion criteria. So most of the patients for attending our clinics were not eligible for the study. So these are the findings from telomere length. So as you can see, telomeres were significantly shorter in schizophrenia and major depressive disorder patients compared to healthy controls, but also to bipolar disorder. The latter group actually showed the longest telomeres of all the groups. And the reason could be related to the effect of treatment with mood stabilizers, because all the patients were under treatment when we sampled them for telomere length measuring. And we also tested the correlation between length of treatment with mood stabilizers and telomere length, and actually found a positive correlation. So these are the findings for inflammatory markers. We specifically measured the high sensitivity C-reactive protein. And as you can see here, levels were significantly higher in schizophrenia patients compared to healthy controls. There was no difference in the other groups. We also tested the correlation between levels of C-reactive protein and telomere length, and found a negative significant correlation, suggesting a potential interplay between inflammation and telomere length. So the higher inflammation, the shorter telomeres. The other marker we measured was TNF-alpha, and there were no differences among the groups. However, when we tested the treatment-resistant major depressive patients, we found no difference in telomere length, but we found significantly higher TNF-alpha levels in these patients, even though the sample was pretty small, with just 10 patients. So the summary of the first study is that telomere length was significantly shorter in schizophrenia and major depressive patients compared to non-psychiatric controls. Treatment-resistant patients had no difference in telomere length when compared to controls, but they had higher levels of TNF-alpha, suggesting increased inflammation. But again, the sample was quite small. The second study was carried out in a much larger sample that included 148 treatment-resistant patients and included 121 patients with major depression and 27 bipolar disorder 2 patients, and 335 non-psychiatric controls. Treatment-resistant was defined, referred to the stage 3 of Tay-Sachs-Brush staging method. All patients were treated with ECT. Illness severity and the outcome was assessed using MADRS before treatment, and the day after they finished the ECT sessions and after 1 month. And we call these 2 time points early responders and late responders. In this sample, telomere length was measured as leukocyte telomere length using quantitative PCR. So as I probably mentioned before, is a much more cost-effective approach and is a very good compromise compared to QFISH. So these are the variables that we tested for correlation with telomere length. As you can see from treatment-resistant patients and controls, only gender and age were significantly different. We included age in the model and not gender, because gender was not correlated with telomere length, while age was, as I will show you in a while. These are the same variables comparing responders and non-responders at time point 1, so the early response. As you can expect, there's a difference only in MADRS T1 scores. And these are the responders and non-responders at time point 2. The only differences we see in terms of variables is MADRS scores, of course, and also number of ECT sessions. But in this case, we'd also tested this variable in the models, and there was no significant contribution. So as I said, we found negative correlation between leukocyte telomere length and age, as expected. And we also reported significantly shorter telomeres in in treatment-resistant patients compared to non-psychiatric controls. However, in terms of response to ECT, we found no difference in telomere length when either comparing responders and non-responders at time point 1 or time point 2, so early response or late response. We also tested the correlation between the other variables. Dividing the samples in bipolar and major depressive disorder didn't show any difference. Essentially, bipolar and major depressive patients showed shorter telomeres compared to controls, but there was no difference between the two groups. LTL was not correlated with MADRS score at any of the time points tested, and not with a variation of MADRS scores among the different time points. LTL was not associated with family history of mental disorders, smoke. We also tested the effect of comorbid cardiovascular and metabolic disorders, and there was no effect, and no effect of the number of ECT sessions. LTL was negatively correlated with BMI when controlled for age, but BMI was not significant in the model when we compared differences between responders and non-responders. So to summarize, we showed that major depressive disorder patients and schizophrenia patients have shorter telomeres compared to controls and bipolar disorder patients. Findings from the first study also suggest that inflammation play a role or might play a role in psychiatric disorder, and that there might be an interaction between inflammation and telomere length, especially in schizophrenia. Treatment-resistant major depressive disorder patients showed increased levels of TNF-alpha, and in an independent study, a larger study, we also reported shorter, sorry, LTL compared to controls. Findings from the ECT study, confirm a previous investigation from another group in a paper that was recently published, where, as I said at the beginning, the authors found no difference between responders and non-responders. So suggesting that telomere attrition could not be considered as a predictive marker of response to ECT in treatment-resistant depression. So these are the acknowledgements of the groups I collaborate with, and of course the funding sources. And I'll be happy to take any questions.
Thank you very much, Alessio, for a great and concise presentation here, which I think is on a really interesting topic. Perhaps I have a question or two for you until we wait, for other people to come in with more questions. One is, you mentioned the comorbidities in the TRD ECT response study, that comorbidities like cardiovascular disease and so forth didn't have any effect. How about in the non-ECT study, in study 1? Did you look at any, I mean, it was a small sample size, but did you look at any comorbidities there?
Yeah, it was actually one of the aims of the study, because the hypothesis was also to look for the contribution of comorbidity with age-related disorders. We found any effect. Okay, interesting. And perhaps the second question in the same vein, in the ECT part of the study, or the second part, you mentioned that ECT numbers didn't have any association. What about specific ECT measures, or some of the physiological measures, like length of seizure, intensity of seizure, and so forth, threshold of seizure? Did any of those relate to that? We haven't tested them yet. Actually, I've showed just the first part of our analysis. It's still an ongoing study, so we're also going to test other features, those that you suggested, but also the potential effect of length of treatment with antidepressants and classes of antidepressants or mood stabilizers that patients were taking before starting ECT. We also have some GWAS data for several of the subjects included, so we are also testing the correlation between some of the GWAS hits and telomere length to see if there's any new hint.
There's actually a question related to that from the audience, which asks whether there has been some research recently involving genetic variants associated with telomere length, and referring to earlier this year to a study on telomere length GWAS, and identified 32 associated variants. So that's, I think that points to the link you just made. To
Right, there's several studies and also meta-analysis suggesting several SNPs associated with telomere length, and we are doing several studies not only in ECT but also in other psychiatric disorders that we usually study to test those SNPs actually in our samples.
Yeah,
with several approaches.
Yes, excellent. Before we perhaps close this part of the session, there was one question coming in a bit late to the previous talk, and that was whether there's any knowledge on the number of ECT sessions related to any of the EVGF levels. So whether that's related to the numbers of ECT. Alessandra, do you know anything about that?
No, there is, in my knowledge, there is no data about that in literature.
Okay, right, thanks. So we've covered that question as well. So thanks again, Alessio, for a great talk.
Thank you.
And for answering these questions. I will now hand over to my co-chair, Peter Zandi, to follow through the last part of this session. Thanks.
I was wondering, thank you, Bernhard. I was wondering, Alessio, if I could ask one quick question before we move on.
Sure.
The correlation with mood stabilizers and bipolar disorder and telomere length was very interesting. Were they all on lithium when you say mood stabilizers, or were there
So we the specifics bipolar patients that were included in the study that I showed, they were on lithium, but not in monotherapy. So all of them had lithium, but also other mood stabilizers. And we also had some schizophrenia patients taking mood stabilizers and also some depressed patients. So the effect was regardless of diagnosis in that specific case. But in another study that we published earlier, patients under lithium treatment had a very significant correlation. There was a very significant correlation between length of lithium treatment, so exposure, regardless of clinical efficacy or response, and telomere length. And this was shown in 2 different studies from our group and other studies. So that really looks like a very strong evidence for lithium effect. Yeah. We don't know if it does anything in terms of modulating the clinical efficacy, because to date only 1 or 2 studies report that telomere length could also predict the outcome of lithium treatment. And we didn't find anything. We tested and we didn't find anything in our sample in terms of response.
Very interesting finding. And thanks again for the interesting talk and to the entire panel. So at this point, we wanted to introduce our discussant for today. I don't think our discussant needs any introduction to this group. Pat Sullivan is, I hope I get this right, Pat, Jurgen Distinguished Professor of Psychiatry and Genetics and the Director and Center for Psychiatric Genomics at the University of North Carolina, and also has an appointment at the Karolinska Institute. And also, as I mentioned in my talk, the co-PI of the grant from the NIMH to carry out the genetics of ECT study here in the US. So we look forward to hearing your thoughts on this work within ECT. Thanks, Pat.
Thank you, Peter, and thank you to the panel for allowing me to be discussing. It's been a really interesting session, and I thank the speakers who came before me. So I've been thinking about I've been involved in depression research for 20 plus years, and I think the problem with going after the genetics for major depression has always been the issue of clinical heterogeneity. So I give a couple of just sort of gendered descriptions there of 6 different people: 18-year-old woman with interpersonal disarray and depression, 42-year-old woman who's being abused by her partner and depression, 66-year-old woman with hypertension, type 2 diabetes, and periventricular schmutz on her MRI, again with depression. And then similar age, but this time for an 18-year-old male who's isolated, asocial, and depressed, a 42-year-old man who's divorced and drinking far too much, and a 66-year-old man who's got some mild cognitive decline. All of these individuals would meet criteria for major depression in a valid and probably reproducible way. But I think, given the differences in these human beings, it's very unlikely they all have the same underlying entity. And if that's the case, then the genetics is likely to be different as well. And, I throw up 3 different title pages from a couple of articles. The first was a PGC related one that Doug Levinson wrote about why we had no significant findings in our first couple goes at depression. And really clinical heterogeneity was a big thing. We got past that in the PGC2 paper from 2018, where we supersized it. We basically managed to come up with a way to come up with a very large number of cases, but a lot of these cases were relatively mild and certainly not severe. And the Kai paper indicated there, some issues that there are issues with that paper, but the general idea that if we expand out to include a wide range of heterogeneity, that might be very useful from some perspectives, but it might also be less useful for others. And that's the thing we wanted to come back and think through. So let's go back a step. We are certainly nearly guaranteed to get far larger sample sizes for major depression by large biobank studies, and that's fantastic. And that'll help move things along in a very useful way. However, because many of these large biobank studies have cases of relatively mild severity, the clinical relevance to the people that are seen by clinical psychiatrists in hospital and tertiary, if not quaternary care settings, the relevance is less certain. And I would argue then that we should be studying the most severe forms of depression and other psychiatric disorders as well. And this is also sort of part of the PGC's core identity. This is something that we can do and that biobanks and 23andMe, et cetera, can't really do, because they certainly get a lot of cases, but they typically are not many of the kind of cases that are seen in clinical psychiatry. So the question I would ask is, how can the PGC foster collection of MDD subtypes of greater severity? And I've been fortunate to be able to work with Bernhard and Peter and many other people, Sam Meltzer-Brody, et cetera, and we've been able to come up with a couple of creative solutions to this problem. The first is for postpartum depression, and the second is for ECT. And in fact, this made it into the PGC 4 grant, which we submitted in February. We had reasonable scores. We're hopeful to get a nice decision on funding, but it's not for sure yet by any means. But our third aim 3C, was using the PGC's global network. We want to promote new studies of highly severe or treatment-resistant clinical cases in order to enrich for causal alleles and to increase our translational potential. And the GEN-ECT study is exactly one along those lines. Other non-PGC examples include the CLAS UK study in Cardiff and the Conlegion study that we've heard about yesterday. For postpartum depression, really briefly, we've shown previously that postpartum depression has higher twin pedigree heritability, 55% versus high 30s for major depression, relatively easy to get cases. We've got this Mom Genes app and program with Sam Meltzer-Brody, and basically you can do the whole study by app in the United States and in a few other countries. You essentially, you download the app, we do consent and phenotype screening within the app, a second consent for DNA sampling and genotyping, and they get sent a spit kit by mail. It's been cheap, it's been fast. We've been able to get a great ad campaign going in this, and we've been fortunate to have been tagged by a couple of high-profile influencers. But for the idea for ECT is basically summarized in this diagram that was done by Louis at Karolinska. Essentially, if we think about a liability threshold model, there's a first threshold that you get past, and if you have that liability above A, you develop MDD. And this may be mild or moderate, but it certainly is not severe. And about 15% of the population, we know for epidemiological studies, will get over that first threshold. But a far smaller proportion will exceed the second threshold. And this is more the phenotype extreme MDD. And a fraction of people, a fair number of people in the second group will get major depression. And this idea was the notion that led to the study, the PGC MDD Gen ECT IC study and the NIMH Gen ECT study being led by Bernhard and Peter. First question is the basic hypothesis supported? And it certainly seems like it. I'll refer to Mikael's talk, but I think this graph sort of captures it, where we certainly see that the SNP heritability is far higher within the Gen ECT, with the prefect ECT population than with the PGC population. In addition, there was a direct comparison against a Swedish case group who were getting CBT for mild or moderate depression, and it appears as if the GRS were higher. The advantages of working on ECT is the patients are easily found. They come to a clinic at specific cases and time. If you can't talk to them the first time, you can often get them the second or third time, because the ECT has multiple events per course. We've noted both in Johns Hopkins and at UNC, there's a high response rate. I think in our pilot study, we had 90% of people we approached consented and gave a DNA sample, which is far higher than any other site. Anecdotally, the patients who are in these studies get it. They understand that there's something distinctly different about them, and they're very pro-genetics in our perspective. Often there's IV access as part of ECT, so it's relatively easy to get a blood sample. And then typically there's a consult note an ECT, a physician has actually put together in a 1 or 2 page summary, all the salient information you'd want to grab phenotypically. So there's a number of reasons why this. And so the 2 broad aims of these things are, first, genetic discovery. If these individuals, as it appears, have inherited a higher burden of common variants, this will be an advantage toward finding for genetic discovery. Second, as an obvious thing to do is, within cases, can we figure out predictors of good versus poor outcome, or good or people that get a lot of adverse side effects or adverse events from ECT? Because obviously those, if that were to work, that could actually be a useful tool in clinical psychiatry. And then in the PGC4 grant, the other thing that's a big part, in fact, it's part of aim 1, is to increase ancestral diversity. There was a great symposium yesterday on this, and it's a huge theme within the PGC right now, is that we really need to cover the world and not just people of European ancestry. And we're doing that in a bunch of different ways, but there's an opportunity right here, right now. So the World Congress this year has many attendees who are not from the typical places. They're not the typical people that go to this conference historically. And many individuals that are probably at this conference have access to severe samples because they happen to work in a university hospital. That's the catchment area for a much larger area. And often those are the places that would be doing ECT. So there's an opportunity here. If a colleague were to be able to do this, to get genetic data, this is an opportunity to get genetic data on samples from your country, your patients, as well as to get the know-how about how to do high quality psych genetics research. The brief idea is that if you were to get, say, 500 cases who are getting ECT for unipolar depression, ethics, consent, a blood sample, and reasonably good clinical phenotypes, we will be able to get them genotyped. The people that work with us on this will become an author and a partner on the PGC MDD broadly, as well as the Gen ECT and genetic specialist studies. And the people involved will contribute their data to the aims of these studies, but will also get back their data, their own genetic data, to use however they would like. And this can also be a wonderful framework to foster other studies. We're just interested in the genetics, but that's only one potential predictor / biomarker. Per Dr. Minelli and Dr. Squassiana's talks, it might be possible to supersize biomarker studies. It might be a way to actually do really large and very carefully integrated studies of various types of other biomarkers, to look at telomeres, to look at imaging, et cetera. And one of the key things is with the combination of genetics and biomarkers, it's possible to use Mendelian randomization to gauge causality. And I note briefly a great example of this down below, the paper that John McGrath did on vitamin D. And vitamin D, of course, lower vitamin D is a risk factor for schizophrenia, but they showed in this paper that in fact it may not be a causal risk factor, but it might in fact be the other way around. And so stuff like that is stuff that we can do where we can actually figure out not only are the biomarkers associated, but also whether there's plausible causality. And that's my discussion. And thank you very much.
Thank you, Pat. That was a wonderful summary of the effort and of its importance of the effort and the promise of the effort. So we appreciate it. We do have 5 minutes before we close. Bernhard, should we open up for any questions for Pat or the panelists, or close here?
No, I think if we still have questions, shoot them out and try to get Pat to answer them.
Yeah, and again, you can put your questions into the chat box if you do have questions. Yeah. I myself have lots of questions, and I'm involved in this interesting effort. And I'll go ahead and jump in until somebody else raises questions. One thing we've heard, the heterogeneity is an important issue, and the whole idea of focusing on the severe end is to try to minimize the heterogeneity.
Yep.
In some of the previous studies, we saw this combining depression from bipolar disorder and unipolar depression. I was wondering what your thoughts are on that. Does it make more sense to broaden our recruitment efforts? We've debated and thought about this. I don't know if we've ever come to a clear answer. I don't know what your thinking is in terms of the relative costs and benefits of broadening to include bipolar.
Yeah. So my general answer, honestly, for 15 years has been the following. For this question comes up all the time. If you go narrow early, you can't actually address your assumption. right? If you have a broader inclusion criteria, provided you can get everybody genotyped, then you can actually ask the question, you can put your assumption to the test. Mm-hmm. Is it in fact different? Are there important differences, or are the similarities greater than the differences? And I think, you know, one of the things that the last 15 years has taught me is that a lot of the things I would have sworn were true ended up not being supported by genetic data. And so I think it's important, you know, if, you know, the trade-off, of course, is you can't go implausibly large, and you have to have the cash to get it genotyped. That's really the key thing. And provided you can actually, you know, stay within, you know, broaden things plausibly, and if you've got enough way, you have some way to get them genotyped, then I think it makes sense to go a bit broader than a bit narrower.
And then test empirically whether there's a difference.
Right.
Interesting. Other thoughts or questions? Yeah.
I have a question for Pat, if I may, regarding treatment refractory patients. So, which I know you're interested in, because I'm thinking if you see a schizophrenia patient, it's being treatment refractory, you can assume that it's a pretty severe case, but I'm not sure it's the same thing with depression. I think many treatment refractory cases are in fact not depression, whereas something else. And that worries me somewhat with respect to ECT, because sometimes it's a last resort treatment, and then you get no effect, and then you conclude that it's not depression, it's something else.
When you say it's not depression, what would it be if it's not? So you've got somebody that looks like they're depressed, meets all the criteria for depression, are lined up for ECT, but you're arguing that there's a subset of people that might not have depression. What do they have?
Yeah, that's a big question. But I mean, personality syndromes is one option, of course, sure. And also cognitive impairment of some sort. Mm-hmm.
I agree with you, but I think we have to start with what we can observe. And given that we don't have great ways for identifying these, essentially one of the reasons we're doing genetics is trying to identify them. So let me spin a couple of plausible scenarios that if one had large data, one could actually look at. First, along the same lines, I might argue that some people who are, let's say we've got a 25,000, the GenECT study wants to get 25,000 people genotyped who are with unipolar depression getting ready for ECT. I'm going to argue that some of those individuals are going to have a large copy number variant. I'm going to argue that, you know, in typical depression, we don't see this, but I'm going to argue that there's going to be a subset of people that are going to have 22q11, 16p11, et cetera. And that might be one subtype that's different. Another subtype is that, of course, is that some people will develop bipolar disorder in the future. Or even schizophrenia. I mean, they're unipolar today, but if you assess them 5 years from now or 10 years from now, they actually would have gone to a different diagnosis. But I think the interesting idea is that, you know, if we could define each person by a panel of genetic, of GRS, so we could get a GRS for personality-related traits, for bipolar, for major depression, et cetera, and then apply some of the new and interesting high data visualization and clustering techniques, we might have a chance to identify those subsets if the ends are big enough and if the division is something that we can look at. So I agree with you that I think people getting lined up for ECT with severe depression, as well as any depressed group, is not just one thing. I think it's more homogeneous, but it's not perfectly homogeneous. But we might have a chance to figure out the subsets in this population in particular.
Bernhard,
and maybe sort of.
Sorry, Bernhard, I'm going to jump in. There's a question from the audience, and I figure we should give the audience a chance, and we have about 1 minute. So sorry, I don't mean to jump in, but Andrew McIntosh asked, what do you consider is the optimal study design for severe MDD? Recruiting consented participants introduces biases related to factors promoting participation generally. Is register-based the best approach? Where's the ideal source of controls in your opinion? Also, issues related to super control status influencing apparent architecture of MDD. So it's a multifaceted question in 1 minute or less.
Who's going to do this one?
We just leave that for people to ponder upon as we close on this session.
Yeah, I mean, there's biases all over the place, right? I mean, Andrew apologizes. No, it's a great question, Andrew. I mean, yeah, I mean, yes, if anytime you have a recruitment stat where somebody can say yes or no, and especially if you're taking people from a clinical source where there's lots of referral biases to get in there, yeah, you always buy bias. The advantage of using a population-based register, like some of the stuff that is available in the Nordic countries, is that it's relatively unbiased. But a lot depends on, who gets identified as having depression, and if they have a diagnosis of depression or treatment for depression, do they actually have it? Because, I think one of my colleagues told me that something like 13% of women in Stockholm were on an SSRI. And I'd be very surprised if half of those people actually had depression per se by a clinical definition. So there's trade-off each way, Andrew. I think the ideal source is to do as many different things as one can and to do them carefully, and then to look at the results across the different study designs with this issue in mind.
And I think with that, we should probably end. We've gone over our time. I wanted to thank all the panelists for some interesting and stimulating talks. Thank you, Pat, for the nice summary and discussion of the issues. And thank you for all the attendees to listening to us present.